Yes, I do feel similar to yeast in many ways. Of course, yeast don't have brains, so humans are more than yeast, but everything that yeast does is also relevant for what happens in our cells.
By deciphering the very fundamentals of a process that happens in a cell, that process is going to lead to the research of how a disease comes about, for example, or how a gene is mutated to cause cancer. When you think of yeast, you actually think of Saccharomyces cerevisiae. In daily life, people think of that one baker's yeast. Enters in bread.
To make wine.
Brews the beer.
Grows on fruit. So it's a very rapidly growing little organism that helps in our everyday lives.
The two species we use, Saccharomyces pombe and japonicus, actually are very, very close genetically. The word pombe actually means beer in Swahili because some scientists actually found it in beer. The other one we use, as you can sort of tell from the name, it was isolated in a strawberry field in Japan.
And we're working on chromosomes, the blueprint of all our lives.
We actually look at the process of cell division.
Budding yeast, it grows by producing a little new bud and then spinning that off.
So you have the cell, that's sort of this sort of shape. They grow to a certain size, and then they divide into two.
And that was an amazing model to look at cell cycle regulation, because a lot of those things are very similar to what we see in humans. A human cell's got about 4 meters of DNA to be packed into a really small micrometer-sized nucleus. But this packaging task, how you can get so much DNA organized, duplicated, and then faithfully segregated, that's the question that we use yeast to address. So yeast is a very useful system for us to study that because it's a lot simpler. We have just one enzyme in place where humans would probably have at least 3. That's, for example, true in cell cycle regulation, but also in chromosome architecture.
It really depends on the field of research that you do. Obviously, people work on mice, people work on zebrafish, people work on frogs, but because the genes are very well conserved between yeast and human, it makes sense to study them in yeast.
The yeast chromosome is a lot smaller, so we don't really see this characteristic X shape that you see in a human chromosome. However, you could imagine a yeast chromosome is just sort of a small part of a human chromosome. But how you then make that into a human X shape, that really is a question for the future.